Beneficial traits of transgenic plants. Transgenic plants have many beneficial traits such as insect resistance, virus resistance, herbicide resistance. A variety of insects, mites, and nematodes significantly reduce the yield and quality of the crop plants. The efforts to produce insect-resistant transgenic plants by using innovative and eco-friendly methods have been quite successful. Insect-resistant plants contain either a gene from the bacterium B. thuringiensis or the cowpea trypsin inhibitor gene. Spores of Bacillus thuringiensis contain a crystalline protein or cryprotein. In the insect gut, the protein breaks down to release a toxin known as a delta endotoxin. This toxin binds and creates spores in the intestinal lining, resulting in ion imbalance, paralysis of the digestive system, and after a few days, insect death. Different versions of the cry genes, also known as Bt genes, have been identified. They are effective against different orders of insects. Bt cotton is the most popular of the Bt crops. It was planted in 1996 and 1997. The Bt gene was isolated and transferred from a bacterium Bacillus thuringiensis to American cotton. The American cotton was subsequently crossed with Indian cotton to introduce the gene into native varieties. Bt gene introduced genetically into the cotton seeds protects the plants from bollworm. A. Lepidoptera, a major pest of cotton. The worm feeding on the leaves of a Bt cotton plant becomes lethargic and sleepy, thereby causing less damage to the plant. Field trials have shown that farmers who grew the Bt variety obtained 25 to 75% more cotton than those who grew the normal variety. Also, Bt cotton requires only 2 sprays of chemical pesticide against 8 sprays for the normal variety. According to the Director General of the Indian Council of Agricultural Research, India uses about half of its pesticides on cotton to fight the bollworm menace. Use of Bt cotton has led to a 3 to 27% increase in cotton yield in countries where it is grown. Several strategies have been used for engineering plants for viral resistance, and these utilize the genes from the virus itself. The first transgenic plant of this type was a tobacco produced in 1986. Which contained the coat protein gene of tobacco mosaic virus or TMV. When these plants were inoculated with TMV, it did not infect the plant. Expression of a virus coat protein gene not only confers resistance to the concerned virus, but also gives a measure of resistance to the related viruses. These plants also showed delayed expression of disease symptoms for tomato mosaic virus or TOMV and tobacco mild green mosaic virus or TMGMV. Weeds are unwanted plants which decrease the crop yields and compete with crop plants for light, water and nutrients. Herbicides are pesticides which are used to kill these unwanted weeds. Herbicides inhibit the function of a protein or an enzyme that is involved in certain biological processes. Several biotechnological strategies for weed control are being used. For instance, the overproduction of herbicide target enzyme, usually in the chloroplast in the plant, which makes the plant insensitive to the herbicide. For example, glyphosate is a glycine derivative and a herbicide which is found to be effective against 76 of the world's worst 78 weeds. It kills the plant by being the competitive inhibitor of the enzyme 5-enoyl pyrovilshikimate 3 phosphate synthase or EPSPS in the shikimic acid pathway. Due to its structural similarity with the substrate phosphonol pyruvate, glyphosate binds more tightly with EPSPS and thus blocks the shikimic acid pathway. Certain strategies were used to provide glyphosate resistance to plants. It was found that EPSPS gene was overexpressed in Petunia due to gene amplification. EPSPS gene was isolated from Petunia and introduced into the other plants. These plants could tolerate glyphosate at a dose of 2 to 4 times higher than that required to. By using mutant EPSPS genes, a single base substitution from C to T resulted in the change of an amino acid from proline to serine in EPSPS. The modified enzyme cannot bind to glyphosate and thus provides resistance. The plants may inherit male sterility either from the nucleus or the cytoplasm. It is possible to introduce male sterility through genetic manipulations. In tobacco plants, a gene that codes for an enzyme barnase which inhibits pollen formation is introduced. This gene is expressed in terms of specific cells involved in pollen production. The restoration of male fertility is done by introducing another gene Bastar that suppresses the activity of barnase at the onset of the breeding season. By using this approach, transgenic plants of tobacco, cauliflower, cotton, tomato, corn, lettuce, etc. with male sterility have been developed.